Hello and welcome to this BioProcess International Ask the Expert webcast. I'm your host, Alex Nicolaitis, the webcast coordinator for BioProcess International. Before we get started, just a couple of notes. This webcast is being recorded and will be made available for replay in the multimedia section of our website. We've muted the audio lines, but we welcome you to type in your questions for our speaker in the question and answer window on your screen. After the presentation, we'll begin the Q&A portion and I'll ask our speaker your questions. Your questions in the Q&A window will only be visible to myself and our speaker. So thank you for joining us today. It's now my pleasure to introduce our speaker, Ray Rendon, Application Development Manager from Colder Products Company.
Hello, everyone. Thank you all for your attendance today. As mentioned earlier, my name is Ray Rendon, and I'm an Application Development Manager for CPC. On today's call, we're going to examine small format tubing commonly found in the cell therapy labs, and we're going to discuss open connections and sterile welds, briefly go through some cell processing procedures, share some voice of the customer, and as a result, we're going to share a new alternative connector with you today. So I do want to put out a quick disclaimer that I have pictures on my following slide deck. They're not mine. I just took them off Google Images after typing in some keywords, so I put credit under each picture. Here's a brief bio about myself. So the open secret within the cell and gene therapy community is that clinical manufacturing, when performed bench to bedside, is being done utilizing small format tubing. With well over 150 stem cell transplant centers in the US, all residing within a hospital or a cancer center setting, single-use supplies with small format tubing attached are entrenched in the hospital setting. So here we have a small list of single-use supplies used within the cell therapy industry. Each plays an important role in the manufacturing of cell therapy products, and each is used at different time points. So to be as brief as we can today, we're just going to examine 3 points where these supplies are used. You have leukapheresis. This is the process of collecting the patient's white blood cells. The manufacturing process alone is pretty elaborate, but we try to simplify it to a few points. You could isolate the white blood cells, you could activate the T cells, engineer T cells with CAR gene, or grow and expand the number of selected cells through cell culture. And really, this is a snippet. There's just so many things that you could talk about in this category. But finally, you have storage and infusion. You're going to have to cryopreserve the cells and store them for later use if you don't plan on infusing the cellular products fresh. So in leukapheresis, this is where we see the collection bags that we mentioned before. In the manufacturing processes, we'll see most of all the other supplies mentioned before. And when it comes time for storage and infusion, we have the cryopreservation bags. So in these examples, we've shown a very raw timeline of events which occurs within the cell therapy lab, collection to infusion, or vein to vein. And single-use supplies with small format tubing attached are used throughout. So a problem that arises with small format tubing is you almost always have open connections. And these type of connections increase the risk for contamination of the cellular product, but also they increase the risk of the operator being exposed to the biologic. So for those reasons, open connections are made under a biological safety cabinet. So very briefly, we want to illustrate the connections made, and we're going to do it with a manual cell process called a volume reduction. So essentially, blood consists of red cells, white cells, and plasma. And since we only care about the stem cells in this process, if we can reduce the volume of plasma, we can likely reduce the burden of processing, and ultimately the volume being reinfused back to the patient. So to begin, we start with a collected apheresis bag, which is holding our starting material of stem cells. This bag has a spike port and a single donor line attached. The apheresis bag is either spiked in a hood to a transfer pack, or the same transfer bag is sterile welded to the apheresis bag. But once connected, the stem cells are transferred from the apheresis bag into the empty transfer bag. So at this point, all we really have is the same thing that we started with. The cells are just inside of a transfer bag instead of a bag off the apheresis machine. The next step is to sterile weld another empty transfer pack to the recently filled bag. But importantly, the weld is kept closed so the cells remain in the first bag. The next step is to place the 2 connected bags into a centrifuge, and you're going to spin them for several minutes, maybe 10 minutes or so. And when it's done, the cells are carefully removed, and you can see the stem cells are now separated into fractions. The bottom layer are the red cells. The middle is the buffy coat, which is full of white cells and platelets. That's where your stem cells are going to be. And the top layer is the plasma. And that's the fraction that we can remove in order to reduce the overall volume. So to remove the plasma, the full bag is placed on a plasma expressor. And when you do that, an acrylic plate pushes against the full bag as shown in the second photo on the bottom. When this occurs, the plasma is expressed out of the bag and into the attached empty bag. So this is the final setup once the cells are moved back into the biological safety cabinet. But once again, notice the small format tubing on the bags, the spike ports, and the lure locks, all posing a risk of contamination. So if no additional processing is required at this time, such as cell selection or cell culture, these cells are ready for storage and cryopreservation. But before we move on, I did mention the use of a sterile connecting device. The sterile connecting device represents a pretty big advantage for the cell processing team because they can achieve closed sterile connections outside of the biological safety cabinet. Additionally, the welder also allows two products to connect to each other, which wouldn't do so normally. And while sterile connecting has a clear advantage in that it helps solve a problem for the cell manufacturing process, in several instances, the welder may actually hinder the progress of the cell therapy industry as a whole. And this is because it allows end users to create their own parts in-house. So instead of an industry supplier creating a specialized part to fit the needs of the cell therapy industry, perhaps two transfer packs connected to each other, cell therapy teams have instead just started making the parts themselves, maybe even utilizing supplies that were not intended to connect in the first place. And this pro versus con effect for welding doesn't just stop at single-use supplies, it goes into automation as well. In some cases, automated cell therapy devices require a sterile connection in order for the machine to interface with the cells, buffer, or anything else at all. Essentially, the machine won't operate as intended without a sterile weld connection being made first, or multiple times. And it's this ability to sterile connect anything to a machine which may lead to operator error, leading to waste or contamination, or maybe a piece of automation even working outside of its scope, creating risk for the entire process, including the developer of the machine. And so with everything that we covered so far, we set out to get input from over 50 industry experts of both automation and single-use supplies to specifically address how open connections were being made and if there were any pain points. So here's some key messages that we heard. We want to simplify production. We want to make connections at any point in the process because, sometimes space is limited and you can't always have a machine right there next to you to help you out. We want to reduce the time needed to make a connection. We want to reduce the need for extra tubing. We want to eliminate the need for ancillary equipment, decrease the risk, of course, of contamination, and of course, we want to make it simple. So ultimately, what we learned, there was some level of frustration with current connection technologies. So with that in mind, we asked ourselves, what is required for a new aseptic sterile connector? Well, we want to de-risk the situation. Open connections, just by their design, have caps that you take off and put back on, or parts that you have to swab. With a welder, there's always a concern about getting a faulty weld. So these are areas of risk that need to be addressed. We also want to streamline the process. The operators at the bench should need less training than that of which is already required of them. Currently, the operator needs to know what parts to use, how to order and maintain inventory of those parts, where to connect them, how to connect them, what additional parts and devices are required for the connection to be made. Any alternative should clean all of this up. And by the way, if we can do this, we're also reducing risk. Reliability. Any new connector should be equivalent, if not better, in reliability when compared to current open technologies. Next are time efficiencies. And any new connection should be equivalent or faster than the time currently needed for open connections or welds. And if we stop to think about it for a second, how long do you think it really takes to make a few open connections? Maybe a sterile weld or two, maybe a spike going into a spike port, twisting a lure lock together. Ultimately, it doesn't take that long. So any alternative has to be faster and better than that. And time efficiencies should not be overlooked. The overriding theme at most cell therapy conferences this year was the ongoing need for operator trainings, including a shortage of operators in general. So any alternative that is faster, intuitive, reliable, needs less training could be tremendously valuable. Next are cost efficiencies. To be fair, there are a lot of moving parts to what makes something cost effective. It's almost relative to how many open connections are you making, how big is your team, how many cellular products are you processing. But when we examined this topic, we found a lot of hidden costs for open connections. We won't do a deep dive on this call, but briefly, you could have the cost of the equipment, the cost of the wafers, cost of service and maintenance, cost of in-house service and maintenance. It just goes on and on. Training, retraining, labor, time, dedicated benchtop space. But essentially, there's always a cost that's intangible. It's not the cost you're paying up front, but the overall cost of everything. And the last one is control. The new alternative connector must give control to the manufacturing lab. So for the operators at the bench, this means no changes will occur in how they currently process their cells. This is very important for the operator, so it's worth repeating. The end user at the bench will see no changes at all in how they process their cells. They retain complete control of their process. For the OEM, or the manufacturer of the cell therapy device or piece of automation, the new alternative should yield more control on how their devices are used, ensuring that there's limited off-label use and closing up any gaps in their risk assessments. Ideally, this added control might even provide a modernization or an update to their existing product lineup. So with all that said, today CPC would like to introduce the MicroConnects aseptic connector. It operates with 3 different sizes of small format tubing, and it is the alternative to the process of open connections and tube welding. To make a connection with the MicroConnects, you simply pinch, click, and pull. So you pinch just to remove the protective cover. You click to join the parts together, and you will hear an audible click when you do this. And finally, you pull. You pull the membrane strips directly away from the connector, thus opening the flow path, and you're done. You have an aseptic sterile connection made outside of a biological safety cabinet, and you're ready for sterile fluid transfer. Now, of special note, on the middle picture, you can see the connectors attach only one way. If you were to rotate a single end, the parts would no longer interlock. So we're making things easy and intuitive for the operators at the bench. The MicroConnects comes in 3 different termination sizes, 1/8, 3/32, and 1/16 inch. The parts are genderless and suitable for gamma or autoclave sterilization. And so here we have just a few images to help show how the MicroConnects could be used in the cell manufacturing process. In each example shown, the MicroConnects represents a drop-in replacement for current open connections. So all we did is we simply replaced the spikes or the luer locks or even just a sealed donor tube with the MicroConnects. Now, of special note, I want to zoom in on the rocker platform and the mini reactor shown. On these devices, they have large diameter tubes attached as well. And these large tubes represent a great transition point for us today. When you have more elaborate cell processing setups, it's typical to find large cell culture platforms such as bioreactors in place or other systems where large format tubing is used as the primary connection point. Here at CPC, we've been servicing the needs of these bioreactors or other large systems with a part called the AseptiQuick. The AseptiQuick accommodates a variety of large diameter tube sizes, and the AseptiQuick offers all the benefits we already discussed today, the MicroConnects. In fact, it can be said that the new MicroConnects is based upon the inventiveness of the AseptiQuick. So today, CPC is in a position to offer sterile aseptic connectors for the entire cell and gene therapy process. For the first time, if you're in biopharma and are processing with large format tubing, you likely already know about the AseptiQuick product lineup. But if you have a need to scale down to small format tubing, the MicroConnects will represent a similar technology package to what you already know. And on the flip side, if an industry trend develops where the clinical cell therapy labs are required to scale up, industrialize, or just reduce the number of open connections made, the MicroConnects represents an option that will accomplish this for them. As a final benefit to using the MicroConnects, cell therapy labs can now begin using a technology package which is aligned with their biopharma counterparts. So that concludes today's very quick presentation. I'd be glad to hang on and answer a few questions. And sincerely, thank you all for your time today.
Okay, great. And as a reminder to our audience, you can go ahead and type in your questions for our speaker now. So our first question is the membrane the same as what is used in other CPC connections?
Sure. So just to repeat, so the question is the membrane the same as other CPC connections? Yeah, that's a good question. So I'll just cover all the components in the microconnects. The body is a polycarbonate and the seal is made of silicone. Now, the membrane itself is a hydrophobic polyether sulfone. But really, the most important thing to note from that is that membrane is what allows the connector to be sterilized by either gamma sterilization or autoclave, high temp autoclaves. So to be more precise about the question asked, we do offer the same type of membrane in our aseptic quick lineup. We call it the high temp version. But if anyone on the call is already familiar with the aseptic quicks, you'll recognize it as the white body with the white pull tabs.
And do the same size connectors only attach to each other?
Yeah, no, that's another great question. So good news, the same size connectors do not have to attach to each other if you don't want them to. So if we take the 1/8 inch microconnects as an example, you could simply attach another 1/8 inch microconnects to it and you're good to go. But if you need to reduce the diameter of tubing, you could absolutely get, as an example, the 1/8 inch half and connect a 3/32 inch half to create a reducer if need be. And the same would work for any combination of the sizes. All the parts work together in the same product family.
And does it disconnect?
So unfortunately, it does not disconnect at this time. We do have the concept in the works for the future, but right now the part is only a one-way connection. To get the versatility that you're looking for disconnect, you would probably need to seal the tube off, or if you're looking for multiple connections, you would probably need an OEM manufacturer to make a manifold set with several connectors attached. So unfortunately, it doesn't disconnect at this time, but we are working on that.
What kind of testing has been done on this part?
So yeah, we absolutely have validation reports which share all the details of all the testing that's been performed. Rest assured, the connectors have been torture tested, so they're good to go. Off the top of my head, some examples of the testing that we've done are, let's see, pressure and temperature testing, burst testing, tensile strength side load testing, freeze and thaw down to -80 Celsius. Of course, biocompatibility and extractable data is available. That's just kind of a quick rundown, but we absolutely do have validation reports that we can share.
All right, well, it looks like we're running a bit short on time, so if we don't get to your question, we'll be passing them along to Ray, who can follow up with you directly. Please go ahead and continue to type in any questions you have, and we'll get those passed along. The last question for the webcast is, are samples available?
Yeah, definitely, and thank you for such a fun and easy question. We would love to send samples. You can contact me at the email on the page right there to get started, or just interact with our website. Either way, we'll likely just ask you what tube sizes you're using so we can send the right parts to you. Worst case, we can send a small sample kit to you with a variety of sizes. We also offer operator trainings, so someone like myself can come to your facility, or we could show you the parts on a one-on-one webinar if that's preferred, but we can absolutely get you samples.
Okay, well, great. Thank you, Ray.
Yeah, thank you, and thanks for everyone who on the call. I appreciate y'all.
And thank you to our audience for joining us. The recorded version of this webcast will be available for viewing on demand on our website, and as a registered attendee, you will receive a follow-up email providing you with a direct link. We look forward to having you join us at future BioProcess International Ask the Expert webcasts. Look for those announcements in your inbox. Goodbye.